All living organisms must be able to respond to their environment. That is one of the characteristics of life that we teach to 7th graders. In order to respond to the environment, cells have to have some way to get the information, to get the signal from the outside to the inside so that it can influence the cell to carry out the processes that will ultimately lead to some sort of physiological response. One of the ways that is done is by the G protein coupled receptors, and that's the topic of our lesson today. So let's look at this question a little more closely. How do living organisms respond to the environment? All living organisms are made up of cells. In this particular illustration here, we have a square of the cell or the plasma membrane here, and upon that plasma membrane and running through it, we have all kinds of proteins. The cell, surrounded by this bilayer phospholipid membrane, is embedded with proteins, and many of these are receptor proteins, proteins that will take a message and send it through to the inside of the cell. Every animal species expresses hundreds of different G protein coupled receptors specifically, and these G protein coupled receptors respond to a wide variety of external stimuli and send that message through to the interior. Let's look a little more closely at what a G protein coupled receptor is. It is a cell surface receptor protein. That means it receives a message. It's on the cell surface and it is on the exterior so that it can receive a message. But it also spans the plasma membrane. And not only does it span it, but it makes 7 passes with its helices through the membrane. And it carries a signal to an intracellular molecule that is right inside the membrane. And that activates that intermediate or that intracellular molecule. And that molecule is the G protein on the inside of the cell membrane. This can also be called a heptahelical receptor because of its shape and the fact that there are 7 of these helical sections of a protein, or sometimes it's called a 7-transmembrane receptor. Let's take a closer look at this structure. These G protein coupled receptors have similar cores. Then they have an extracellular N-terminus, and then they also have the intracellular C-terminus on the inside. The top of the page would be the outside of the cell. And then you can see the 7 alpha helices that are spanning the membrane. And we know that the exposed elements of the receptor molecule actually serve as ligand binding elements, and that some types of the receptors have a form of pocket between the helices on the exterior side. And some of those pockets are deep, and some of them form near the surface, depending on the type of protein it is. They also have the transmembrane helices number 5 and number 6 can actually move outward, creating a cavity on the cytoplasmic side, and that's what serves as the docking site for the G proteins. How prevalent are this type of receptor? Well, we've already said that each animal species expresses hundreds of different GPCRs. The human genome encodes about 1,000 different types for 1,000 different purposes, and they are found in the cell membranes of mammals, invertebrates, plants, and microorganisms. So these are receptors on the cell. So what kind of signals do they receive? Well, they can receive signals from the outside, like some of them receive signals from light waves in our eyes, in the rods of our eyes. Others can receive odorants, so smells. Some of them receive hormones as signals, and neurotransmitters as signals, and some of them molecules that are lipids. An example of a signal molecule that we will be talking about today is epinephrine. As an example of G protein coupled receptor signal transduction, we're going to use the body's response to fear. And we've already said that one of those signals is epinephrine. Epinephrine is also called adrenaline. Fear causes the adrenal glands to produce adrenaline, and this is the fight or flight chemical and that response that we feel when we're suddenly frightened. The beta-adrenergic type of G protein coupled receptor is there to bind with the adrenaline, and you'll notice that the G protein is attached to the bottom of that receptor, or is close by. And the G protein is all of those parts. It's trimeric. It has 3 parts. So you'll see alpha, beta, and gamma parts to the G protein. Those are all part of the G protein. Now it's called G protein because it has that guanine nucleotide. When the glucagon binds with the receptor, there is a change, a conformational change in shape. And that change in shape has an effect on the G protein. There's enzymes that catalyze the opening of the nucleotide binding pocket in the G protein, and the GDP is released, and GTP flows in from the cytoplasm. It's abundant in the cytoplasm, and it flows right in. Now, all of these conformational changes change that G protein's affinity for its parts there, and we'll see that in the next slide. But you see in this slide that the GDP is released, and the GTP is now going to be bound to the G protein. So let's go to the next slide and see that. In this slide, we're going to take a much closer look at what we just talked about. You see the adrenaline molecule, it's just attaching to the receptor there. You can see the GDP on the tan subunit, and that must be the alpha subunit because that's the one that the nucleotide is attached to. And then the blue and green are the beta and the gamma subunits. And you'll see from the first to the second diagram there, the GDP has been exchanged for GTP, and the alpha subunit of the G protein has combined with the adenylate cyclase, and you'll see that there is being produced cyclic AMP. So what is happening there is adenyl cyclase catalyzes ATP to CAMP, and CAMP is a second messenger that can be used throughout the whole cell for many different processes. So now that we have cyclic AMP, we're ready for our next step in this receptors in action. Okay, now we can finish up. The adenylate cyclase catalyzes the ATP to CAMP with the help of that GTP. And the cyclic AMP is called a second messenger. And that second messenger will go throughout the cell and be a part of a lot of different processes in the cell. That increase in cyclic AMP in the cell leads to the activation of another enzyme called protein kinase A. And protein kinase A has a big job of phosphorylating enzymes, and those phosphorylated enzymes are then activated, and they can produce their physiological effects. So in this diagram, you can see that some of the effects of protein kinase A are to produce glucose from glycogen And also in the case of adrenaline, to promote muscle contraction, because remember in the fight or flight reaction, one of the things you'll need to do is use your muscles, and to increase your heart rate so that you'll have enough oxygen in order to fly away or to fight. G-protein coupled receptors are a part of signal transduction, and those G-protein subunits, they bind with effectors, and some of the examples of the effectors are ion channels, ion pumps, enzymes, which we saw, the G-protein, the alpha G-protein bound with an enzyme that was able to change ATP into AMP. They can also take part in transcription inducing, so they can cause transcription to occur. And the effectors will then carry out those cellular processes that lead to that physiological response. And it all started as a signal to a G-protein coupled receptor.